The trench that breathes. Research log. Dr. Lena Ruiz, Chief Biologist, RV Hadal Dawn, Expedition, Day 14. Depth 9,212 meters. Location, classified, pending institutional review. We've named the site Lazarus Point. We named it that because finding stable bedrock at this depth feels like a resurrection. Most of the abyssal plain is soft sediment. Ooze, really. The accumulated remains of everything that's died in the water column above and drifted down over millions of years. You try to anchor a research station to it, and the whole thing tilts and sinks and becomes a very expensive mistake. Stable porous bedrock at 9 kilometers down, that's rare. That's once in a career rare. We anchored Trench Sentinel to it 4 hours ago. She's holding beautifully. Let me introduce the team. For the record, Marcus Webb, structural engineer, the one who spent 3 months designing the Sentinel's anchoring system and has been insufferably smug about it since we hit stable lock. Dr. Yuki Tanaka, marine biologist specializing in abyssal fauna, who hasn't slept more than 4 hours since we started descent because she keeps finding things she's never seen before. James Okoro, our pilot and certified deep-sea medic, Navy-trained, 12 years of saturation diving, the calmest human being I've ever shared a submersible with. And me, biologist, expedition lead, the one responsible if any of this goes wrong. We're down here for one reason: abyssal gigantism. I want to take a moment to explain that, because if you've never heard the term, you're about to understand why this depth class of ocean is the most scientifically extraordinary environment on Earth. At the surface, animals have evolutionary pressure toward moderate size. Too big and you're inefficient. Too small and you can't compete. The deep ocean throws that logic out entirely. Down here, below 4,000 meters, and especially in the hadal zones of the deep trenches, animals grow to sizes that have no parallel anywhere else on the planet. Giant isopods, crustaceans related to the pill bugs you find under garden rocks. At the surface, they're a centimeter long. In the deep trenches, they reach 60, 70 centimeters, the size of small dogs. They scuttle across the sediment in the pitch dark at 9 kilometers depth. Colossal squid, Mesonychoteotis hamiltonii. The largest confirmed specimens exceed 14 meters. Eyes the size of dinner plates, tentacles lined with rotating hooks. Larger by mass than any creature we have confirmed living in the ocean today. Tube worms at hydrothermal vents, 3 meters long, thriving in water hot enough to melt lead, in complete absence of sunlight. We don't entirely understand why the deep makes things big. Current theories involve the cold, metabolic slowdown, longer lifespans, more time to grow, the pressure, the nutrient density of certain deep currents. But there's a school of thought I find more compelling, and it's why I pushed for this expedition. What if the trench itself is a nutrient superhighway? What if the geological structure of certain deep formations creates conditions for biological scaling that we haven't yet mapped? What if there are organisms down here that we haven't found simply because we haven't been willing to sit still long enough to look? The Sentinel is going to sit still. For 30 days, anchored to Lazarus Point, we're going to listen. Marcus keeps calling the anchor formation the biggest stable spot we've ever found. He's not wrong. The sonar returns on this formation are extraordinary. Porous, dense, extensive. The exact geological composition we'd want for a long-term anchor. I asked him this morning how large he thought it was. He said, "Big, very big. We'll map it properly once we've settled." We haven't mapped it yet. There's been something else occupying our attention. A minor instrument anomaly that Yuki flagged during our first watch cycle. A low rhythmic pulse in the water around the station. Roughly one cycle every 14 seconds. She initially logged it as a localized current eddy. The trench topography creates strange flows, and the Sentinel is a blunt object in a complex fluid environment. But I've been watching it for 4 hours now, It isn't random, it's regular. 14 seconds in, 14 seconds out. Almost like breathing. It's almost like breathing. I'm logging that as instrument artifact. Probably a resonance frequency in the anchor cables. Marcus will check it in the morning. The station feels settled. That's the word. Like it found where it wanted to be. We're going to sleep now. First full rest cycle in 36 hours. End log. Research log, day 15. 0340 hours. I couldn't sleep. Yuki was awake when I came out of my bunk. She was at the sample station, and she had the look she gets when something has contradicted her model of the world. Very still, very focused. The kind of attention that means she's running and rerunning an observation because it refuses to resolve. She'd been analyzing the core samples from our anchor drill. Porous basalt, we'd assumed. The kind of mineral formation you get from slow volcanic activity at depth. She showed me the microscopy. The outer centimeter, maybe 2 centimeters, of the core sample is mineral. Calcium carbonate with some basalt inclusions, exactly what we expected. But below that surface layer, the sample changes. The material is organic, not fossilized, not ancient biological material that got mineralized over time. That happens, we know how to identify it. This is fresh organic tissue, dense, fibrous, arranged in a structure that Yuki described as broadly analogous to connective tissue in large vertebrates, but not vertebrate. And running through it, she had to zoom the microscopy to maximum to show me this. Running through it were channels, vessels, ranging from millimeters to roughly a centimeter in diameter, pulsing. Slowly, regularly, 14 seconds in, 14 seconds out. The pulse we'd been reading in the water around us wasn't a current eddy. It's in the rock, it's in the formation. Whatever is moving through those capillary channels, it's creating a pressure differential large enough to register on our external sensors. Yuki thinks it's nutrient transport. She said, very calmly, very professionally, that if this formation is biological in origin, then the channel system represents a circulatory analog, that the formation may be essentially alive. I asked her what kind of organism we were looking at. She said, something we don't have a classification for. At 0600 hours, Marcus ran the full sonar mapping he'd been postponing. He's been very quiet since. He came to find me in the sample lab at 0800, and he set a tablet in front of me with the sonar composite rendering on it, and he said, Lena, I need you to look at this and tell me I'm reading it wrong. I looked at it for a long time. The formation we're anchored to doesn't end. The sonar returns extend to the limit of our mapping range in every direction, 12 kilometers north, 9 kilometers south, approximately 7 kilometers east before the signal degrades against the trench wall. And those returns are consistent. The same density, the same porosity, the same internal channel structure throughout. We are anchored to something that spans, at minimum, hundreds of square kilometers, possibly much more. Marcus said very quietly, that's bigger than Manhattan. That's bigger than any geological formation I've ever worked with that didn't have its own topographical category. I didn't tell him what Yuki found in the core sample. I needed another hour to think. At 1100 hours, something happened that I'm logging carefully because I want the record to be precise. The 4 of us were in the main lab for a briefing. Me, Marcus, Yuki, and James. I was explaining what Yuki had found. I'd gotten through the organic tissue, the capillary system, the pulse rate. James, who monitors physiological data for the crew as part of his medical duties, checked his wrist unit. And then he checked the external sensor feed. And then he said, very carefully, does anyone else notice that our heart rates are averaging 14 beats per minute apart from each other right now? We all checked. He was right. The 4 of us, sitting in the same room under the same conditions, had heart rates spread across a 14 beat per minute window. James said, 14 seconds, 14 beats. I don't know if that means anything. I said I didn't know either. The comms have been echoing strangely since yesterday. A half-second delay on internal ship-to-ship communication that our system shouldn't produce, like the signal is traveling through something it shouldn't have to travel through. And Yuki told me, just before this log, that when she was collecting the secondary core sample this afternoon, she had the distinct, specific, persistent sensation that something was watching her. Not from the dark above, not from behind, from below, from the formation itself. End log. Research log, day 15, 22:30 hours. We had a seismic event at 1900 hours. Not large, magnitude 2.1, localized, centered directly beneath the station. At any other site, I'd log it as minor tectonic activity and move on. We're in a deep trench. Geological instability is expected. But the tremor had a specific character that James noticed, and Marcus confirmed when he reviewed the seismographic data. It didn't propagate outward the way a tectonic event does. That ripple, that wave spreading from an epicenter, it was discrete. It originated at one point, moved in a specific direction, and stopped, like a muscle contraction, like a twitch. We also found the vents. There are 12 of them that we've mapped so far, distributed across the formation surface within our sensor range. We'd initially catalogued them as hydrothermal vent structures. Not uncommon, but they're not emitting heat. They're emitting particulate, a fine, dense cloud of microscopic material that disperses slowly in the water column. Yuki collected a sample. I'm going to read from her analysis verbatim, because I don't trust my own paraphrase to be accurate. Particulate sample from Vent 4 is dense with unclassified microorganisms. Morphology suggests motility toward biological tissue. Specifically, the sample exhibits chemotactic behavior, consistent with organisms evolved to penetrate living cellular structures. They appear to be seeking ingress. They want inside. Yuki wrote, "They want inside" in a scientific log. I didn't edit it out. At 2100 hours, Marcus reported that the seal around his secondary suit glove, the one that he'd been using for external sample retrieval, had developed a rash along the contact line. Red, raised, geometric. Not the irregular pattern of an allergic reaction. Precise. Almost like a print. He said it itched. He said the itching made him think, very specifically and without being able to explain why. Of hunger. Of needing to be fed. He said he'd taken an antihistamine and gone to his bunk. At 2140, I ran the drill. The core drill on the Sentinel is rated to penetrate 2 meters of dense rock. I set it for maximum depth and ran it into the formation directly below our anchor point. I wanted to know what was down there. I wanted confirmation or refutation of what the shallow samples were suggesting. At one point, 3 meters, the drill hit something that stopped it cold. Not rock. The resistance profile was wrong for rock, too yielding at initial contact, then suddenly and completely resistant, like hitting a dense nerve bundle, like hitting a nerve. I was still reviewing the drill telemetry when the station moved. Not swayed, moved. A single, massive, coordinated shift. The entire Sentinel lurching 30 degrees off axis and then steadying. Every alarm on board triggered simultaneously. James was running the length of the corridor before I'd processed what had happened. Through the main viewport, I watched the formation below us. The surface crust, that 2-centimeter layer of mineral material, was cracking. Not randomly, in lines, in organized directional lines that spread outward from our anchor point in both directions, following what I now understood were the paths of the largest internal channels. And beneath the cracked crust, where the mineral layer fell away in plates, light. Bioluminescent tissue, brilliant blue-white, pulsing rapidly now, much faster than 14 seconds. The pulse was accelerating, responding, like a reflex arc, like the involuntary firing of a nervous system that had just been irritated at depth. We'd hit a nerve. We had literally hit a nerve. Yuki came to stand beside me at the viewport, and we watched together in silence for approximately 40 seconds. Then she said, very quietly, "Lena, I need to tell you what I think this is." I said, "Tell me." She said, "I think the trench is the body. I think the trench itself is the organism. The walls, the floor, everything we've been anchored to and sampling. I think it's all one thing, one continuous biological entity. And I think the abyssal gigantism we came here to study, all of it, the isopods, the squid, the tube worms, I think they're not just adapted to the environment. I think they're part of it. I think they're cells." She paused. She said, "And I think we are parasites. Very small parasites on a very large animal. And I think we just hurt it." I looked at the viewport. The bioluminescent veins were spreading. Not just below us. I could see them extending outward along the trench wall in both directions, lighting up in sequence like a signal propagating through a nervous system, like news traveling, like an immune response being organized. My tablet pinged with a satellite data update from the surface monitoring array. Trenches worldwide, the Mariana, the Tonga, the Kermadec, the South Sandwich, all of them showing synchronized seismic activity within the last 40 minutes. All of them pulsing on the same frequency. Not 14 seconds, faster now, like a heartbeat that's been startled. End log. Research log, day 16, 0015 hours, emergency record. I'm making this log because I don't know how long we have, and I need a record to exist somewhere. Evacuation protocol initiated at 23:10 hours. Reason, the station is being consumed. That is not a metaphor. The formation surface has been rising, tissue expanding, flowing upward and around the station legs in slow, deliberate movement. We first noticed it on the external cameras as a discoloration around the anchor points. By 2250, it had reached the lower equipment bay. By 2300, it was at the viewport line on the starboard side. It isn't attacking us. I want to be precise about that. There is no aggression in it. It is simply growing around us, enveloping us, the way skin grows over a splinter, the way a body deals with something foreign and irritating and small. We are the splinter. James got the emergency ascent pod prepped in 17 minutes. The pod is rated for 4 people, but the organism has closed around the primary launch tube. The tissue is dense enough now that the tube won't clear. Marcus has been trying to cut through it with the emergency laser cutter. He says the tissue responds to the laser the way soft material responds to heat. It contracts, and then it regrows, and the regrowth is faster than the cutting. At 23:35, Marcus stopped cutting. He looked at his hands. The rash on his wrist had spread, past the glove line now, up his forearm, still geometric, still precise. And he said, "I can't feel my fingers on this side. I can feel something else. I don't know how to explain it." I know how to explain it. The spores from the vents, the microorganisms Yuki identified as motile toward biological tissue, the ones that want inside. They've been in the cabin atmosphere since we started collecting samples. We've been breathing them. We've been breathing them for 12 hours. Yuki confirmed it clinically, professionally, with the same controlled tone she's used throughout this entire expedition. The spore load in cabin air is significant. The organisms are adapted for integration into living tissue. Given exposure time, I would expect them to have established preliminary contact with epithelial tissue in anyone aboard. She paused. I would expect mild cognitive interference, a sense of connection to the host organism, possible auditory artifacts. I asked her what kind of auditory artifacts. She said, voices, probably. Very simple, repetitive, I've been hearing something for approximately 2 hours that I've been categorizing as equipment noise. A low, slow sound that resolves, if I focus on it, into something almost like words. 2 words, over and over. Feed me. James found the solution at 0000 hours, not the primary launch tube. That's gone, buried in tissue. But the emergency beacon ejector on the station's upper dorsal surface is still clear. It's rated to propel a 40-centimeter signal buoy to the surface. James spent 19 minutes modifying it. He can fit 1 person in the ejector housing if they curl small enough. 1 person. He looked at me, and I looked at him, and we both understood what that meant. And neither of us said it out loud. Marcus was already showing signs of cognitive integration with the organism. He'd stopped speaking, stopped responding to his name, was sitting very quietly against the wall with his eyes open and his expression perfectly calm. Yuki had begun her final logs with the systematic care of someone who has accepted a situation and wants to leave complete records. She told me to go. She said, "Go, Lena. Tell them what this is. Tell them it's real. Tell them we are not the apex of biological scale on this planet. We are a recent development on the surface of something that has been here since before we had bones. Tell them it's not aggressive. Tell them it's just indifferent. We don't matter to it. We never did." I said her name. She said, "I can hear it now. It's not as bad as you'd think. Go." James put me in the ejector housing and sealed it from outside. The launch was violent. I blacked out for some portion of the ascent. My wrist unit logged 6 hours of travel time from that depth to the surface, which means I went through proper decompression stages on automatic. The pod is designed for that. I don't remember any of it. What I remember is waking up on the surface to grey morning sky and the sound of a rescue helicopter, and then a lot of very bright lights and people asking me questions I wasn't ready to answer. I'm answering them now. They found what I said they'd find in my right forearm. A density on imaging, irregular, biological, approximately 3 centimeters in the subcutaneous layer. The doctors want to remove it. I've asked them to wait. Not because I don't understand the risk, because when I'm very quiet and very still, in the space between sleeping and waking, in the moment before the mind reasserts its walls, I can feel it pulsing. 14 seconds in, 14 seconds out, and I think about what Yuki said, that it's indifferent, that we don't matter to it, that we're lice on its hide. And I used to find that thought terrible, but I've been watching the surface monitoring feeds from my hospital bed. Every major ocean trench on the planet is still showing synchronized seismic activity. The pulse is still accelerating. It hasn't stopped since we hit that nerve, since we drilled into that cluster and the formation convulsed and the bioluminescent veins lit up like a nervous system receiving bad news. Something changed in it. Not aggression, not hunger, awareness. Something 7 orders of magnitude larger than us. Something that has been resting in the dark since before our species learned to walk upright. Something just noticed that it has passengers. We are still here. We have always been here, on its surface, in our boats and our nets and our submersibles, drilling small holes, dragging small samples, making small noise. And it never noticed before. It notices now. 14 seconds. Feed me.